Hello everyone, my name is Ana. I'm a master's student in oceanography from University of São Paulo in Brazil, and I appreciate the opportunity to talk here in AbGradCon 2021. Today I'm going to be talking about the chemosynthetic production in extreme temperature gradients from Deception Island, Antarctica, and its implications for the limits of life. The chemosynthesis is the process where organisms obtain the energy used in the synthesis of organic compounds from chemical sources through redox reactions and not from the usual sunlight radiation. It is a process that can be dominant in systems with a large input of reduced compounds, used as electron donors, as the hydrothermal vents or cold seeps. Chemosynthetic organisms can be highly diverse in metabolism, phylogeny, or habitats, which made a lot of chemosynthetic microorganisms to be also extremophiles, living in harsh environmental conditions. The Deception Island, located in Antarctic Peninsula, is one of these extremophile environments, and a unique habitat on Earth. It is formed by a volcanic cone that collapsed with the ocean invading the caldera. Therefore, an active geothermal point with marine influence. There are active fumaroles reaching values of 100 degrees Celsius, whereas around 60% of the island is covered by glaciers. The interaction between ice, ocean, and volcanic activity creates pronounced temperature and biogeochemical gradients in a small distance, favoring the growth of metabolically diverse extremophilic microorganisms and allowing all temperature-adapted microorganisms as hyperthermophiles, thermophiles, mesophiles, and psychrophiles coexisting and possible interacting in the same environment. All these characteristics make Deception a possible analog of other Earth environments, not for the environment itself, but rather for the parameters that can be limiting for life as well as chemosynthetic production in a planetary context. For example, the cryovolcanoes in Enceladus or the extinct volcanoes in Mars. The main goal of this study was to understand the limits for life and for chemosynthetic production in one of these parameters, in this case, the extreme temperature gradients. So, the samples were collected in 2018, Austral summer, during the 27th Brazilian Antarctic expedition in the scope of Microsphere project. We collect sediment samples in triplicates from two geothermal sites on Deception Island, Antarctic Peninsula, called Waller's Bay and Fumarole Bay. As you can see on this map, in each one of these geothermal sites, it was defined a transect containing 3 points, one for a glacier, here represented by the blue color, and two from fumaroles, the yellow and red ones. The fumarole samples of Waller's Bay reach 60 and 53 degrees Celsius, and the fumarole samples of Fumarole Bay were hotter, reaching 97 and 98 degrees Celsius. The glacier points in both locales are reaching around 0 degrees Celsius. The depth of the sediment recovery was approximately 5 centimeters, and the distance between fumaroles and glaciers at each site were approximately 15 meters. After the sampling, we performed in situ simulated dark carbon fixation incubations with C14 sodium bicarbonate for a period of 36 hours in 3 temperatures, for 60 and 90 degrees. After the incubation period, the samples were exposed to hydrochloric acid to remove the remaining carbon, and then they were transferred to vials with scintillation cocktail and left in the dark for 12 hours before the reading counted through liquid scintillator per 30 minutes. The results given in disintegrations per minute were converted into production rates of chemosynthesis. As a result, we observed that the DCF rates varied from 0.07 to 0.69 microgram of carbon by cubic meter by hour in Waller's Bay, and from 0.12 to 1.79 in Fumarolo Bay. So the highest DCF values were found in Fumarolo Bay, where are localized the hottest fumaroles which display high hydrogenous sulfide emissions. When you look at the average of chemosynthetic production, we got some interesting results. In this graph, the colors represent the temperatures of incubation. So blue is 4 degrees Celsius, the yellow is 60, and the red is 90. And we found 0.27 of chemosynthetic production in a glacier sample from Waller's Bay when incubated at 90 degrees. Conversely, we found 0.16 of chemosynthetic production rate in Fumarolo sample from Fumarolo Bay when incubated at 4 degrees Celsius. We also found that the glacier sample from Waller's Bay WBC from a glacier present more chemosynthetic production when incubated at 6 degrees Celsius than when incubated at 4 degrees Celsius. The stranger high chemosynthetic production in WBC could be due to a process of dispersion of the microbial cells in the environment, allowing the presence of microbes adapted for a hotter temperature in this sample. And as I mentioned before, the highest chemosynthetic production rates do appear at Fumarolo Bay. But more specifically, we observe such rates in the sample recovered from the glacier sediments when incubated at 4 degrees Celsius. So, the environment of a polar volcano also favors the chemosynthetic production from microbes living in the ice and snow, which shows the huge importance of these ecosystems not only for the microbial communities living near the source of reduced compounds, but also for the ones more distant. Unfortunately, the dark carbon fixation analysis gives us the information about the process of chemosynthesis and not about the taxonomy of microbial communities present in the samples. So to better understand why we found these chemosynthetic rates, more analysis is necessary, and this will be the future steps of this project. We also performed the statistical analysis in the data and ANOVA between the temperatures of incubation and the t-test between fumarole versus glaciers, that is the environment, and the t-test between the sample's location, Fumarolo Bay versus Waller's Bay, considering a significant difference, a p-value less than 0.05. With this, we noticed that there were no significant difference between the environment nor in the incubation temperature. However, we do observe a significant difference when compared the sample's location, Fumarolo Bay versus Waller's Bay. This could be due to the presence of hottest fumaroles in Fumarolo Bay that give the local more chemosynthetic production rates, as we had already seen before. And also the temperature in environment or the temperature of incubation were not significant parameters for the chemosynthesis in these geothermal points. Therefore, we conclude that extremely low and high temperatures were non-limiting factors for the chemosynthetic production. Furthermore, the geochemistry of a volcanic polar environment selects active chemosynthetic communities at both marls and glacier sediments, and besides that, the dark carbon fixation analysis could be a simple and novel tool for life detection on astrobiology research. In the future, we plan to perform the metagenomic and metatranscriptomic analysis to discover which microorganisms compose the microbial communities in each one of the samples, and which of them are the actual responsible for the chemosynthesis process that occurs here. I would like to thank my collaborators and also thank the Microsphere Project, the Antarctic Brazilian Program, the University of São Paulo, the Oceanographic Institute, and the Microcean and Lecon Labs for all the support given to realize this project. Thanks so much for watching.